So our next speaker is Zach Grinspan, who is going to give us an update on the phenylbutyrate trial for STXBP1 and some other DEEs.
Thanks, Jim, and thanks again for having me speak at the conference. Thanks to the organizers. These are my disclosures. I have an AI disclosure. I did use a little AI for data cleaning and analysis. I wanted to just put that out there. And actually, before I start, I wanted to just say a word of thanks to the community and for your patience with the study. I keep coming back year after year, and I get little nuggets of updates. So for those of you who heard me speak before, this will be very similar. For those new, I did want to make sure that I gave some background to the study, what it is. I do have some updated results, and all the data is collected. We have everything in hand, and we're now working actively on making tables, figures, and writing. I start with my thank yous. So the story of this trial is really one of community support and love. This was started as just a concept for someone who had not done clinical trials, and then it was small amounts of funding from patient-led advocacy groups, from STXBP1, from Clara Inspired, from the Orphan Disease Center that got it started, and then once we started and got onto a roll, we then got some money from pharma to make it go forward. And then the work I'm going to show you is a collaboration between our center in New York and with the center in Colorado at Children's Colorado, which has been a real joy. That collaboration has been very fruitful, and I wanted to make sure that those shout-outs happened early on. So the drug I'm going to talk about is 4-phenylbutyrate. It's a drug that was FDA approved already for a different disorder, for urea cycle disorders. It sops up ammonia from the periphery and lowers serum ammonia in people who have these disorders. It crosses the blood-brain barrier. It comes in 2 flavors. There's a sodium phenylbutyrate that's very salty and disgusting, and then there's a glycerol phenylbutyrate that came out later, which is much more tolerable. It's a liquid. It doesn't taste too bad. The reason we tried this is that there was an accumulating evidence of preclinical data suggesting that this could work for monogenetic disorders in children. The top paper is from our center. Jackie Bure is the senior. Noah Guberson was the postdoc that did the work. That suggested that this might work in MUNC18-1, which is STXBP1. And then as we got started, we learned that there was additional evidence for several other disorders. So STX is the one that we all know, but it also seemed to work preclinically for SLC6A1. There's some work in GABA-A receptor disorders. We haven't given it to people with LGI1, but there's some evidence it might work for that disorder. SLC6A8 is creatine transporter deficiency. There was not preclinical data, but we had an inkling that it might work for SYNGAP, so we did try them. And then there was some preclinical data in Dravet syndrome. The organizing principle with this group of disorders, they're all associated with epilepsy and seizures. They all seem to impact synaptic function. And other than SLC6A8, they're all heterozygous disorders, so one good copy, one bad copy. One question that I often get asked is, well, why do you think this drug would work? And the different mechanistic explanations for why it might work kept shifting. The more people I talked to, the more different ideas people had. So we have found some efficacy, and so the question is like, why? And the way I've thought about this is you've got some cellular mechanisms that then converge on some kind of functional impact. So here are 2, 4, 6, 8, 8 or 9 different cellular mechanisms, all of which have some evidence or some rationale to think that it might be helpful. But at the end of the day, either you're stabilizing wild type, you're rescuing some mutant, you're protecting from a toxic effect from a mutant, or there may be some other compensatory thing that this drug is doing that's not listed in those cellular mechanisms. It may, for example, just be a traditional anti-seizure drug that doesn't necessarily have anything to do with the genetics. Regardless, we were motivated enough by the preclinical data to launch the trial, which is registered here. What we did was an open-label, multi-dose, single-treatment group trial at 2 centers, ourselves in New York and in Colorado. We did a screening call. We collected some baseline data about seizure frequency. People would come in for a 3-day, 2-night EEG, come back between 6 and 12 weeks later for a second EEG, and then we gave people the option either to wean off the drug or to stay on. We have extended this trial again and again. December 2026 is for sure the end, but we've continued to follow folks, ideally once a year in person and then with periodic telehealth visits. We published some preliminary results in 2024, I think. So this is out as a preprint, which talks about our first 20 patients. And the manuscript we're working on now will talk about 50. These are the 50 that we enrolled, 10 with STXBP1. There was an early signal favoring SLC6A1, so we ended up going to 12, 9 SYNGAP, 7 with Dravet, 7 with GABA-A receptor subunit disorders, and then 3 with creatine transporter, and then 2 one-offs where we had some inkling maybe it would work for these other 2 genetic disorders. We designed the study really as a safety and tolerability study. We certainly measured efficacy, but the safety and tolerability data was very, very wonderful. The main thing that we discovered, that we found, was that there were no new side effects that the FDA did not already know about. So that was very reassuring to us that there wasn't something unexpected in the DEE population that could lead to problems with this medicine. I use the word wonderful, which is the wrong word. We did have some serious adverse events. The main issue with this drug is metabolic acidosis, particularly in kids, in the sort of the toddler age, and some of the younger kids. Often we get a metabolic acidosis, and that led to several kids having to go to the hospital, so we had some hospitalizations for that. In a couple kids, the acidosis was quite refractory, and it was only by lowering the phenylbutyrate that we were able to get control of it. Many kids, though, who were liking the other beneficial effects of the medicine, lowered the drug, added things like Bicitra in order to manage the acidosis, and many of them stay on it. I wanted to say, so of the 50 who enrolled, 38 are continuing on the medicine today. There were 12 who have stopped for a variety of reasons. Most of those, 10 were for efficacy, and then 2 of them were for both efficacy and the severity of the side effects. So this is our main outcome. This has been vetted multiple times. This is not AI. I read all of these charts. And these numbers have changed a little bit over the past year as I've gone into the charts, reviewed the seizure diaries, and we also, compared to prior presentations, we decided to be very firm about what was the seizure outcome we were measuring. So this is 50% reduction of seizures at that first follow-up. For the first 20, that was a 6-week follow-up, and then we extended that to align a little closer with more standard anti-seizure drug trials at 12 weeks. So in the STXBP1, there's 9 people who we felt we could assign this outcome, and 5 of those 9 met this outcome of 50% seizure reduction. The big story for the trial was the SLC6A1, where 9 out of 10 met this outcome. And then several of the other groups, we had some responders. In SYNGAP, 6 of 9, GABA-A, 3 of 7, in Dravet, SCN1A, 2 of 7. A note about Dravet, the non-responders really didn't respond. There really wasn't any question of like, well, is it a little? And so we had been pessimistic about that. But these 2 who responded, they're real responders. And so I think one of the things we need to figure out is what is the basket? Like, what is the proper basket to think about this drug for? So our total responder rate across the 50 is 57%. And then there are 4 where we just didn't feel like we could interpret the result reliably. These are the STXBP1 outcomes in some more detail. I think in prior years I've said 6 6 responders. The first non-responder did have meaningful reduction, about 25%, 26, 30% reduction. So at the 50% level, we have to put them in the non-responder. But I did want to highlight that the family was quite happy with the results, even though it didn't meet the outcome. Other things that I would comment here, let's see. It was a mix of mutations, so we had microdeletions, missense, and truncation mutations. The seizure type varied. The one seizure type that really clearly did not respond were epileptic spasms. So many STXBP1 kids present or have epileptic spasms early on. That is classically a very difficult seizure type to control without things like steroids, vigabatrin, ACTH. And so those kids did not respond, but the other seizure types often did. You can see that short term, we had two children who were seizure-free. One of those then reverted and began to have monthly seizures by a year, and at the last visit was still having them. Another one largely continues to be seizure-free. That child had one breakthrough seizure, but that's been it. You can see there at months of exposure at the last visit, is what that column header is supposed to read. We've got some kids who have been on this drug for 4 years or more. And so from a tolerability standpoint, a lot of families are having a good experience with the medication and have been satisfied with its results. And then the other interesting thing is we've seen in other sessions what the natural history of the seizures are. Often seizures will sort of burn out in this population, that they have seizures early on and then they kind of go away. So one of the children who had very difficult to control epilepsy early on, the last time I spoke with that family, the child had been seizure-free 6 months. I'm not going to attribute that to phenylbutyrate, but it does highlight sort of the intersection between a trial and the natural history and how to make sense of the results. We did have one new paper that did come out, so this is exciting. Kristen Barber was a medical student and a resident and fellow with us at Cornell, and is now she did a genetics fellowship at UCSD, and she's faculty there now. She got on the phone and spoke with I think 18 different kids around the world who had taken phenylbutyrate in one form or another off-label, so not people in our trial. The way this visualization works is on the x-axis there is sort of frequency of seizures, and then the blue arrows demonstrate which direction your overall frequency seemed to go after you started the medicine. And so the blue ones that go from 20 per day, et cetera, down to less often are the ones that responded. It was a mix of STX and SLC6A1. So just looking at the STXBP1, you can see that there are more responders than not. And the main story with this paper is that this corroborated our experience in the trial, that many kids who took this drug did find this positive change in their seizure frequency. I want to talk a moment about development. We did measure development, and for those of you who think all about development all the time, kudos. This is very complex data and takes a while to kind of wrap your head around and think through. As a clinician, the way I'm thinking about developmental data is like this. I sort of think about it in this space with chronological age on the x-axis, age equivalent on the y-axis. A neurotypical child grows along the diagonal. As they grow in age, their age equivalent scores and all the developmental measures grows with them. So those are the green dots. Most children with monogenetic DEE plateau. At least that's the clinical experience. That it seems like they get to some certain level of development, and after that, roughly their level of development doesn't change. This is not to say that the small changes that I asked a question about earlier aren't real, aren't valid, and are experienced by the family, but the clinical impression is often that the child roughly is doing about the same stuff that they were at the last visit. And so the goal with any treatment, we would love to get kids back to the neurotypical line, but more likely, treatments will get kids better than they would have been. And so lifting them off of that plateau and getting them to have more positive development. I was a math teacher before I went to medical school, and so I couldn't resist doing a little bit of math with this. So you can write down an equation. This is one example of maybe half a dozen different equations you could use to imagine what this developmental plateau is. I've taken that equation and fit it to data from Simons and from RareX for STXBP1, SLC6A1, and SYNGAP. This is Vineland data, and these plateau curves roughly do seem to fit. The normal dotted line comes up pretty faint. I think you can see it on the screen. And so you can see that this idea of plateau does seem to work, at least conceptually. Is this the right equation? Should we be doing something different? My equation is one parameter. Do we need more than one parameter? I'll leave that to the future. But for now, this is sort of a rough way for us to think about development. This did not reproduce as well as I had hoped. So these are our gold standard in-person developmental assessments in the trial for all 50 kids. The blues are the responders, and then the orange are the non-responders. And what the reason I'm showing you all of this is to highlight that the STXBP1 children, their development is quite impaired. This audience knows that. But this raises all sorts of questions of how do you measure developmental progress when children's level of function is at the lower end of most of the scales. So we can zoom in a little bit, and you can start to see some of the trajectories over time. There are some outliers here. There's some people whose trajectories seem flat. There's some people whose trajectories seem to be lifting a little bit. At a first pass, this is a first pass preliminary, I took that developmental curve, the thing that I did in my high school math teacher capacity, and then looked across 4 domains for 3 diseases to see, did children beat their expectation by 3 months or more in any of these domains? When I did this calculation, I also took a look at the Vineland scores to try to get a good sense of, on average, how are people doing. And so we did see some improvements in the SLC6A1 group and the SYNGAP, but in the SLC6A1, SYNGAP, we saw some improvements, but in STXBP1, we really didn't. And this aligns with my clinical intuition as well from following these kids for so many years, that the gestalt was we got nice control of the seizures. We seemed to do better in that regard, but development felt like people were at their plateau and we weren't really lifting them off. So this aligns with the clinical intuition. So limitations, small sample size. We only have 10 from STXBP1. 50 is nice, but it's a basket and it's from lots of different disorders. It's open label. Many kids had tonic seizures, which are hard to count at night. Some of the kids in the other disorders had absence seizures, which are uncountable and also a difficult outcome. And the natural history, the stuff that you'll hear about with the STAR study, is incompletely characterized for most of the DEEs in our basket. We are continuing to crunch the other non-seizure outcomes. We're working on manuscripts. From a supply access standpoint, so the regulatory and the patent landscape for this drug is very complicated, and I won't go into all the details. Suffice it to say, in the US, there are now 6 generics. And so the list price for a bottle of glycerol phenylbutyrate, which is 25 cc's, which for many kids is not even a week's supply, that was $4,500 before the generics have come out. Now I think there's 2 generics that are selling that bottle at $500 a bottle. It's not cheap, it's not, but it does reduce the burden either to families who want to pay out of pocket or to their insurers from hundreds of thousands of dollars a year to tens of thousands of dollars a year. So we're thinking that's going to help us with access. I've certainly written it off-label for many children, and insurers have paid. There's starting to be conversations about a European trial. The European regulatory environment is incredibly complex. There are some European countries you just write for it and they give it to you. There's others where It takes many, many levels of approval, and others where you just cannot prescribe off-label unless the national health system says that it's approved for that indication. And I think there's some countries where it's not available at all. So that's being discussed. It's a repurposed drug. And so there's also this complication of who funds a trial where you don't have a pharmaceutical sponsor that wants to invest in it, and we know how expensive clinical trials can be. So this piece is very much an ongoing story that we're likely to hear more about in the years to come. The other thing in the US that we are working on, and we haven't had success yet, for Medicaid and for children, if you write enough letters, somebody's heartstrings will get pulled and they'll approve it. We've seen that again and again in multiple states. And so we've been able to get this for kids. It's often a struggle. But for adults, Medicare is very strict. So we had a young man who got glycerol phenylbutyrate, a different disorder, got glycerol phenylbutyrate, got the best seizure control of his life, and then Medicare said, this is not indicated, and we don't see it anywhere in our compendia or anything like that, and so you can't have it. Dad's a lawyer, Dad takes Medicare to court, and Dad loses that lawsuit. So in order for the full range, the full lifespan for all of these disorders to get this drug in the US, we'll need it to be in one of these compendia. There's at least 4 of these organizations. What they do is they list different drugs and their indications both on-label and off-label. So like the cancer community has been extraordinarily effective at getting off-label indication for many of their drugs. We have submitted to all of these. We did that in the fall. And then with Kristen's paper out, and once ours gets out the door, we'll be submitting again. So this, again, an ongoing regulatory issue. Our hope is that the data will be compelling enough that they'll list it in their compendia. I don't see a path at this moment for label extension with FDA. But from that standpoint, this HHS has been asking for comment on repurposed medications. And I know that this drug is on their mind, so we may hear more about how the federal government in this country thinks about repurposed medicines and what those approval processes look like. So that's an ongoing story. Thank you again for having me speak. Thank you for the funders. Thank you for the participants and the families. I got to say, this is a theme that's come up again and again. Being in a clinical trial is hard. So for those families who have schlepped to New York and schlepped to Colorado, we know that it's tough, and it's a big burden on families to do this, and I'm just incredibly grateful for your participation. Thank you. Okay. Did I go long? Was that too long? Oh, I think she was here first. Go ahead.
I do have 2 questions, but I can limit it to 1. I know that the trial itself will be ending in December, the availability to get medication, but do you have any plan to have a type of protocol in place for patients who are on a chronic anhydrase medication or the ketogenic diet to proactively start a citrate medication to help avoid any of the metabolic acidosis? So I would make that a clinical question. I didn't say this, but you're right. Kids who are on keto and this, or on topiramate, the acidosis has been tougher. I think as a clinician, I didn't have screening labs, because we didn't know, but as a clinician, as I've been prescribing this, I'm getting baseline acidosis, I'm following that, and a very low threshold to start something like Bicitra. So clinically, yes, please, have I protocolized that? No. Okay, thank you.
So, quick question. I don't know if you heard me speaking yesterday, but what we saw is with NMN, we actually saw an improvement over 2 years on the motor behavior in Vineland questionnaire. And I saw that you didn't see. So the question is, how old are the children that you are looking at? And maybe we can compare notes.
Yeah, and I want to reply thoughtfully. So many kids, we do see some improvements. And the question is, how is that different than natural history?
Exactly.
And so And so I've been conservative in how to interpret that, often looking at multiple measures to make, and the clinical notes to make a determination like, have they actually gained milestones, and how do we make sense of that? So,
So we compare it to natural history,
Yeah.
like of historic natural history, but it might be interesting. Did you look at very young kids? I mean, how old were the kids?
it's a range. The earliest, there were 2 infants that we enrolled, and then up to, I think, 11 was the oldest at enrollment. So it's a range of ages. And then we know, like, a 4-month-old is going to make some early milestones, but a 10-year-old might not.
So what I'm saying is, if when you feel that you can share, it would be very interesting to kind of compare.
100%.
Thank you.
So our publication strategy is to get a preprint out as soon as there's something that's like coherent, and that while we work on getting it into a journal. So and we're trying to get as granular data in that, whether either it'll be in the figures or as supplemental data, because we very much want everyone to have access to that data.
So I mean, we will do the same.
We're
Thank
yours.
you very
We're
much.
yours. Yeah. Oh, thanks.